Welcome to today's lesson. This is biology lesson for grade 12. Student, in our last lesson, we have seen some genetic clause and the law of segregation, as well what by means monohybrid clause, how we can deduce phenotype in the genotype of a given clause. In our today's lesson again, we will see a Mendelian model of heredity. Student, at the end of this lesson, you will be able to draw a Punnett square for genetic clause, and again distinguish between what is homozygous and heterozygous, and find genetic clause for a dihybrid clause. Well, during his clause activity, Mendel used a given model called Punnett square. Mendel crossed homozygous purple flower with its recessive. He predicted F2 generation that consists 3-4 purple flower plants and 1/4 white flower plants with a phenotypic ratio of 3:1. Here, he takes this one. There is a character that comes from where? From a whiteness one. There is a character that comes from that of a purpleness one. Then after, Mendel result of this cross, all offspring of F1 generation become heterozygous. You can use it as hybrid because one trait comes from that of the purple one, another trait came from that of the white one. They came from different locus. If a trait came from different locus, we call heterozygous rare. In case heterozygous, all F1 generation, F1. Then again, which we call this all F1 generation become all purple here because the character of whiteness doesn't expressed here. When both heterozygous F1 individual become crossed together, always the three this-like trait become observed. Here, P capital, homozygous purple flower, P capital, P small, which we call heterozygous, also purple. The character of P small is not expressed in this manner because it's recessive. And again, finally, P small, homozygote again, which we call white flowers obtained. It is a constant outcome of Mendelian if both homozygous dominant, homozygous recessive cross together. In F2 generation, the ratio of dominant to recessive, here, this one represents dominant, whereas this one represents recessive, is 3:1 always. If certain number given for you, you can find the number of dominant, you can find the number of recessive. Again, in F2 generation, the genotype of this cross is always, you can deduce simply 1:2:1. 1 PP, homozygous PP, and again 2 heterozygous P capital, P small, and again 1 homozygous P small, P small. It is constant for any other 7 contrasting traits. All 7 contrasting traits of Mendel can be follows the same principle here under monohybrid cross only. Well done. Test cross to mean a means of identifying whether a dominant character is homozygous or heterozygous. Identification of homozygous or heterozygous dominant in a given cross. This is done by crossing with its recessive trait. For example here, if P capital, homozygous purple, and again there is P capital, P small, which is heterozygous purple. If this both character can be stand for purpleness, suppose we are uncertain, simply dominant purple character is given for us. We are certain whether the character is homozygous or heterozygous. To check whether it is homozygous or heterozygous, we can cross with their recessive one. we can cross with that of the recessive. If all character become heterozygous, we mean we are certain that it is homozygous dominant. If there is a mixture of recessive one, we are certain that it is heterozygous. For example, here, there is P capital, crossing with P small, then all P capital, P small, there is all P capital, P small, all P capital, P small, all P capital, P small. When you say dihybrid cross, it is a breeding situation that obtain or evolve two contrasting traits. During this one, since two contrasting traits found here, the separation of one trait is independent of another. By that, this law is considered as law of independent assortment. During a monohybrid, during a formation of gamete in a cross, law of segregation is found, separation of gene into allele. But here, law of independent assortment. One gene is assorted independently of another without affecting another gene, in absence of genetic linkage only. Gregor Mendel breed a plant with that of homozygous round yellow seed, which is a dominant, over homozygous plant wrinkled green seed, which is recessive. Remind the student, there is two character here, not one trait. Here, there is a seed shape, which is round, wrinkled. There is again a seed color, which is yellow and the green. He wanted to be crossed together, following the same procedure of the earlier cross or monohybrid cross. And they wanted to be understand what is the progeny of F1 and F2 together. We can apply the monohybrid cross here together, but we can use another here. Let here a parental generation here, a round yellow seed, round seed shape, and again yellow seed color, and again wrinkled, which is a recessive of round, and again green, which is a recessive of yellow, which is homozygous round yellow and homozygous wrinkled green. Here, the gamete for this one can be RY and RY here. Gametes. And again, a gamete for this one can be RY and RY. He self-pollinates them together again. During F1 generation finding, he self-pollinates and find the offspring of F1 generation, whether it is pure or not. Then round seed shape with that of, and again yellow flower color, wrinkled seed shape with that of green flower color here. Then again, if we cross this one together, all the gamete is this one, R from one gamete. I told you that one gamete from that of a dominant one, from that of a recessive one. When R cross this one, R capital, R small, which is that of a monohybrid. Remember that for that of a monohybrid here. Let's remind you again. We are speaking about that two monohybrid. Homozygous round, which is a dominant, homozygous wrinkled, which is recessive, monohybrid. And again, there is homozygous yellow, which is a dominant, crossed with that of a homozygous green, which is recessive. Two monohybrid come together. Now we are speaking about that of two monohybrid when we are speaking about that of a dihybrid. The separation of one trait from the else never affect each other because they are found as a different locus, different position. Then one gamete, else gamete, one gamete from recessive, else gamete. When R capital, this one is from the dominant one, this dimension is from recessive one. The character of recessiveness become masked by R capital. The character of Y small is become masked. By that, the character is said to be round yellow. The character of round yellow here. Round yellow here. Means the character of wrinkled green is masked in F1 generation. Just like that of the monohybrid one, the character of wrinkled and green is become masked. Phenotype of this cross. 100 or all, 100% all round yellow. Remind student, there is another character that is not observed here. Because of the dominant 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 here. Genotype. Genotype of this one, all or 100%. R, Y, small here. All hybrid. Hybrid. He starts from that of homozygous, but the outcome is hybrid in F1 generation. So, Mendel proceed to the next one. Here, all the F1 plants are hybrid, which we call round yellow. That shows the character of roundness and that of yellowness is dominant over wrinkled and the green. This law is considered as the second law of Mendel, which we call law of independent assortment. One gene is inherited independent of another because they are positioned on different locus. If roundness about seed color separate independently from that of seed shape, it never affect each other. This is very key reason for a genetic variation. During a meiosis activity, creation of variation among an organism. This independent assortment of gene, according to the hybrid clause, second law of Mendel is only in the absence of genetic linkage. For example, R may be linked in this manner. In the absence of linkage. If linked together, they cannot be assorted independently. They can move together. And again, it may be R1 and again R1. These two characters cannot be considered as two. Rather, it is considered inherited as single. By that, all the allele become separate or segregated independently in absence of genetic linkage. So, gene can be linked. The phenotype and the genotype of this cross can be calculated in absence of genetic linkage. Good. Then he allowed this hybrid genotype to be self-pollinated. Be sure there is one monohybrid here, there is another monohybrid together here, there is another monohybrid. By dihybrid, two monohybrid come together. So here he find all the genotype of the F1 generation of dihybrid crosses, all hybrid, all round yellow. The gamete of F1 is all gamete of F1 gamete. Here RY and RY cross together. And again there is from recessive RY and again RY here. If R small come here, it is round. And again, if R capital here, it is round again. If Y capital come here, it is yellow. If Y small come here, it is yellow again. Because the character of dominant Y or yellow become dominant this one. And again, the character of round here become dominant here again. The one, if R capital come found here, and again it is round. If R small come here, it is round again. If the only thing that can come around after this one is green. So here, it can give round green. We are come to find how many round green here. The only thing that comes after this one is R here, R with this one. And R with this one. If Y come here, yellow. If Y small come here, yellow again. And again, the another is R with this one again. There is another gamete. R, the only thing that come here is Y. So, the gamete for F2 generation, one F1. This one is F1. This one is another F1. The gamete for this one is RY. R capital Y small, R small Y capital, and again R small small. This one is for another F1. About 16 crosses found here. Here, from here it started from R capital homozygous round yellow. Which I mean, R capital come here, here R capital, and again Y capital here. Within this gamete, there is R capital, there is Y capital, this one. And again, we can cross here. We can cross here. From this one, round yellow can be found around 16, 9 out of 16. And again, the one that looks like this one, round green, can be around 3 from the cross, 316. And again, R small, this one, wrinkled yellow, about 316. Which we call the recessive one, which we call wrinkled green, can be obtained about 116. Generally, if we cross two homozygous dominant with two homozygous recessive character under a dihybrid cross, we can obtain 9, which is round yellow. This one, which is round green. This one, which is wrinkled yellow. This one, which is wrinkled green. It is possible to get the same ratio from the product of two monohybrid cross. Let I remind you again. Here, there are two monohybrid cross in this dihybrid one. Means a cross between round and the wrinkled. Homozygous round and the homozygous wrinkled. Homozygous round, which is dominant, this one is recessive. And again, for circular here, there is homozygous yellow and again homozygous green. So two monohybrid come together to form a dihybrid. Under here, the phenotypic ratio for this one in F2 generation is 75% round and again 25% wrinkled in F2 generation. And again, genotype ratio for this one is again 25% R capital homozygous, 50% R capital small heterozygous or hybrid, 25% homozygous, which is this one R small. The same is true for that of this one again. If we cross these two, we can obtain genotypic in the phenotype of this cross. Thus, if we multiply two monohybrid phenotypic ratio, we can obtain dihybrid phenotypic ratio. For example, here, the end product of phenotypic ratio in F2 generation during monohybrid cross can be 3-to-1 ratio. 3 for dominant, 1 for that of recessive, and again 3 for dominant, 1 for that of recessive again. Let me cross them together. 3 round, 3 yellow, which become 9 round yellow. Then 3 round with 1 green, which can give about 3 round green. Then after, 1 wrinkled with 3 yellow, which can give 3 wrinkled yellow. Finally, 1 wrinkled with 1 green, which can give again 1 wrinkled green. Again, if we multiply two monohybrid genetic ratio, we can obtain the same genetic ratio for dihybrid cross again. We can obtain 1/4 R capital, 1/2 R capital small, and again 1/4 R small, and again 1/4 Y capital, 1/2 Y capital, Y small, 1/4 Y small. Finally, we can obtain this like result of genotypic ratio in F2 generation. This is the indication of two monohybrid can come together to form dihybrid phenotypic in the genotypic ratio. When we say dominance, it is an ability of certain trait in a given allele that can capable to express itself in a phenotype by masking the character of recessive allele. When we see homozygous R capital and R capital small, we say it is round because the character of recessive R small is not expressed. But here, we cannot be simply say it is dominant and again it is recessive. One that can be determined as is, we can depend on what type of dominance we are speaking. Dominance is an ability of allele to be expressed itself in a given phenotype. There are different types of dominance. The one that we are going to speak is complete dominance. When we say complete dominance, when both homozygous and the heterozygous are phenotypically identical. For example, here the character of T capital T small and the T capital T capital, which is heterozygous, which is homozygous, it is phenotypically tall. And again, Y capital, Y small, Y capital, Y capital. This one is homozygous yellow, whereas this one is heterozygous yellow. Because the character of Y small is masked here, it can be said to be yellow. And here again, this one is heterozygous round, R capital R small. This one is homozygous round. Because the character of R small is completely dominated by R capital, we call it is round. So here, the character of T capital totally or completely dominating the character of T small. The character of Y capital is completely dominating the character of Y small. The character of R capital is completely dominating this one. By that, we mean complete dominance. When allele come from dominant character completely dominating allele from recessive one, we call it is complete dominance. Well done. The other type of dominance here is incomplete dominance. Most type of incomplete dominance is mostly observed in flower color expression. Here, during incomplete dominance, when heterozygous character show an intermediate trait of both parents, it show an intermediate character of dominant and intermediate character of recessive one. For example here, there is a cross between homozygous red flower color, which is represented by R capital, with homozygous recessive white color, which is represented by R small. Here, when we cross this one, R capital red and R small white, which finally give R capital R small in F1 generation. In F2, when we cross R capital R small with that of R capital R small, this one, we finally obtain one RR capital and again two RR and again one RR. During complete dominance, the character of homozygous R and heterozygous R can be phenotypically identical. But here, this intermediate character, there is a parental genotype here, we call red here, and again this one is again white here. This show an intermediate character of dominant and the recessive, a pink color. For example here, in F2 generation, R capital R small cross with R capital R small. Then one R capital R capital, two R capital R small, one R small. Or 25% red, 50% pink, and again 25% white. During incomplete, the heterozygous shows an intermediate trait of both parents. This is called incomplete dominance. The another type of dominance is codominance. When we say codominance, it is a pattern of dominance where both allele can be contribute to the phenotype. The character of both allele can be expressed here. This type of example may be most probably observed in human blood group inheritance. We human can have ABO blood group, which can classified into 4 blood types. Accordingly, there is A blood group, there is B blood group, there is AB blood groups, there is O blood groups. Blood can be transferred from donor individual to that of the recipient one. Here, an individual with blood group A may have A blood group here. It may be heterozygous A and the homozygous A. It can have its own antigen, A antigen, and again B antibody. Whereas B blood group can have, which is called homozygous P and the heterozygous P, it is own antigen and reverse A antibody. Where AB blood group can have, it can have AB antigen and again none of antibody. If an individual with AB blood group maybe encounters certain accident, this type of individual can receive blood from any blood type. By that, an individual with AB blood group are considered as universal recipients. But here, O blood group, it can have none of antigen here. And again, AB antibody here. By that, this individual can be considered as universally donating and but receive from O blood group only. By that, O blood group is considered as universal donor, whereas AB blood group are considered as universal recipient. Such type of dominance is said to be codominance if both allele can be contribute to the phenotype. If homozygous red bull gets bred with homozygous white, the patchwork appearance of both white and red color can be found. Means the color of redness, red bull, and the color of white calf become expressed. There is a red found here, there is a white. By that, this calf can be show a patchwork appearance of both white and what? Red one. By that we call codominance. Both allele, which is from red, and allele from that white can be expressed here. Well done. Student, in our today's lesson, we have seen how Gregor Mendel used a Punnett square. And again, we have seen how Mendel can be identified. Certain dominant can be homozygous or recessive, homozygous or heterozygous, by crossing with its recessive, by using the technique of backcross. And again, we deduce what by means dihybrid cross and the law of independent adjustment, how he deduced genotype and the phenotype ratio in F2 generation. In our coming lesson, we will see the means of cell division. Until that, goodbye, students.